Okay. Good morning, everybody. The day second of this symposium, we will start the keynote session 4. The theme is the biological interface and microbiome. The chairperson of this session is Professor Nobuhiro Takahashi, Tohoku University. Thank you for your time.
Okay. Can you hear me? Okay.
Yes.
Okay. Good morning, everyone in Asia, and good evening in everyone in the United States of America. Now, I'd like to start the session of Keynote 4, Biological Interface, Microbiome. I'm Nobuhiro Takahashi, a professor, Division of Oral Records and Biochemistry, Tohoku University, Graduate School of Dentistry. I will take a chair for this session. As you know well, the microbiome is now getting important more and more as a partner of our human body in terms of health and disease. And many researchers around the world are trying to analyze and signify it from various points of view. Today, I'm pleased to invite 4 world-leading experts of oral microbiome research to this session. So I'm very happy. I'm very looking forward to their cutting-edge research and their thoughts on microbiome research and their future directions. Today, due to the limitation time, I'd like to accept questions and comments from the audience via our chat system. And then if time is available, we will have the 10 minutes Q&A and discussion period after this session. So now I would like to introduce first speaker, Dr. Shu-Song He from the Forsyth Institute, USA. Dr. He is currently associate member of staff in the Forsyth Institute and also member of faculty in Harvard School of Dental Medicine. His research on the microbiome is very diverse, including study on bacteria and other microorganisms. And his talk today will build on his excellent series of work. Now, I'd like to invite Dr. He. The title is Oral Microbiome, Beyond Bacteria. Dr. He, please.
Okay, so can you guys see the slides?
Clearly.
Okay, great. Thank you, Professor Takahashi, for the kind introduction. And first of all, I would like to thank the symposium committee for the invitation. It's really a great pleasure to be here and share with you some of the recent progress in the oral microbiome research. It's my hope that by the end of my talk, you will have an overall view of the oral microbiome research, its history, its current state, and its future direction, as well as some of the challenges that we'll be facing. And like many words derived from the omics family of the terminology, microbiome become one of those buzzwords, right? So it signifies the ecological community of the commensal, as well as some of the pathogenic microorganisms actually share our body space and play a very important role in our health and disease. And in a way, the field of oral microbiology start because of a long-term suffering from one of the oral infectious disease, dental caries. It begins with the infamous legend of worm. And in ancient times, many believe that it's the tooth worm that drill a hole through your tooth. And a toothache was simply the result of a worm wiggling within the tooth. And this idea was so popular and it spread through the century and several culture, and with evidence of the tooth of worm and found all over the world. And so this is the first evidence of the tooth worm, and it's in the Sumerian text around 5000 BC. And it was also found in an oracle bone script in 1500 BC, and which is the earliest known form of the Chinese writing. So for example, in this case, as you can see here, so you do have this represent the worm, and these are the teeth, and when you put together, this is actually a Chinese character and for the dental caries. And lastly, this is another example. This is 18th century ivory carving actually found in the France, and it's titled The Tooth Worm in Health Demon. So And however, the revelation of the true identity of the toothworm had to wait for another 6,000 years. And Anthony Leeuwenhoek, and which is considered the father of the microbiology, and he used the single lens microscope and of his own design actually. And for the first time, he observed the tiny worm in his own oral cavity. And the microbes sketched in his notebook are now known as some of the most abundant bacteria residing in the oral cavity, and including this cocci, and as well as the fusiform bacteria. So this fascinating observation, even at the beginning of the birth of the microbiology, had already signaled the complexity of the oral microbiome community. And in 1920, Dr. Clark, he first isolated the streptococci mutant from the human dental caries lesion. And later on in the 60s, and by using the animal model, it was showing that to be able to cause the dental caries. And for a longest period of time, the microbiologists took a reduction approach to try to study the individually isolated bacteria from the oral cavity and try to analyze them individually, and hoping that by understanding this small component, which is individual bacteria, and they trying to piece together what's happening in the oral cavity. And however, it has its limitation, as we all know. The culture-dependent technology cannot really recover all the diversity in the oral cavity, right? And so furthermore, the bacteria that behave in terms of the gene expression, and now we know that could be very greatly affected by its surrounding bacteria. So in a way, there's a need to really better understand the bacteria from a community or ecological perspective. So the breakthrough in technology can often revolutionize the research. So for the microbiome research, it's the DNA sequencing technology. So the first sequencing technology was developed in 1977 by Sangers, as you all know. And he is from the Cambridge University. And it was first commercialized in 2002. And then came the next generation sequencing technology, or NGS, and which is also known as a high throughput sequencing technology. And this sequencing technology will first appear as the Roche 454 technology and was introduced in 2005. And these technology actually are be able to produce a sequence with very high throughput and at much lower cost than the first sequencing technology. So for example, for the human genome, if we use Sanger sequence, it's going to take 15 years, over $100 million. But if you use the NGS technology, it only can take 2 months and $1 million. So however, the second generation of the sequencing, it has its limitations because it can only sequence very small DNA fragment and generate millions of the reads. So it requires really high computing resources. And then came this, the PacBio and the Oxford Nanopore. And these are the third generation of the sequencing technology. And that also classified as a single molecule sequencing technology because they're able to generate this long reads at a much lower cost and very fast. And this NGS based metagenomics and metatranscriptomics, and combined with other new high throughput technology platforms such as the metaproteome and the metabolome, which are basically analyzing all the protein and the small molecule within the community. And these analyses really allow the researchers to study the complex oral microbiome in a very sophisticated and comprehensive, and in many cases, even in a culture independent manner. And in 2007, NIH initiated the Human Microbiome Project, as you all know, which is called HMP. And the first phase frequently referred to as the HMP1 and focus on categorization of the microbiome of the healthy human subject at 5 major body sites, including the oral site, using the 16S and the metagenomic shotgun sequencing. And then there's this second phase, which is referred to as the Integrative Human Microbiome Project, or IHMP. and it generates good resources to really allow the comprehensive characterization of the human microbiota to further our understanding of how this microbiome can impact the human health and disease. And what I want to point out is that at that period of time, and so there's researchers at the Forsyth Institute, they built the first human oral microbiome database, and which is called HOMD. And so it became one of the most visited oral microbiome database. And now it has an extended version, which is called EHOMD, and it includes the nasal as well as the skin-associated bacteria. And we're also going to add the bacteriophage as well as the fungal species into this database. And there are also this new emerging technology, and which really expand our ability and allow better understanding of human oral microbiome at the ecological level. And here I just give you some example. So the first one is spectral imaging. So this is developed by Dr. Jessica Welch from the Marine Biology Laboratory, and also Dr. Gary Borisi from the Forsyth. And the spectral imaging allow you to simultaneously label multiple different bacterial species, and then to get more accurate spatial information that related to the complex community structure. And then the second is the metapangenome. So this is a representation of the totality of the gene that belong to a specific bacterial species that identify in multiple metagenomic sampling of a particular habitat. So by comparing the metapangenome of a specific bacterial species from different habitat, it actually can allow you to identify the gene function that allow this specific colonization in certain habitat by specific bacteria. And the third technology is the targeted isolation and cultivation of what we call yet to be cultured bacteria by reverse genomics. So this method was developed by Dr. Polder at the Oak Ridge National Laboratory. And using this method, they can develop antibody against the cell wall components of the bacteria of your interest based on the genome information. And they can achieve targeted labeling and isolation of the bacterial species. And it can really, facilitate our bacterial isolation and expand the library of the cultivable bacterial species. So all these advancements in the technology really provide valuable tools to allow us to better understand the establishment, the development, as well as the maintenance of the oral microbiome. So this is a elegant study by Dr. Ann Griffin from Ohio State University. So her team examined the bacterial acquisition and the community establishment so over the first year of life. And they provide a very dynamic landscape of the microbial development in the oral cavity. And also, it actually can have a huge impact on the disease prediction, diagnosis, and prognosis. So for example, in a recent study, and they show that by identifying the distinct microbial dynamic between the disease onset and the progression, they can actually come up with a predict model and to predict the future. For example, in this case, it's the caries onset. And it is well established the oral cavity is home to several billions of bacteria. And the dysbiosis of oral microbiome and the imbalance of the host microbial homeostasis can cause all kinds of local disease, such as periodontal disease and dental caries. And meanwhile, the concept of the oral cavity is the source of a distant infection, which is known as the focal infection theory. So it has been debated for at least a century. And more and more evidence actually showing that these oral organisms actually can, potentially indirectly responsible for several systemic disease, such as the cancers, the cardiovascular disease, as well as Alzheimer's disease in this case. So the relationship between the periodontal disease and the systemic disease has been under extensive investigation right now. So far, I have been mainly focused on bacteria due to their dominance in the species richness and abundance and well-established association with oral and systemic disease. But we know that in the oral cavity, it's a well-organized community with very diverse microorganism composition. And in addition to the conventional bacteria, which has been the center and focus of the research in the past, there are other very important microorganisms. For example, there are a lot of the fungal species, which is a collection we call microbiome. Okay, and not to mention all those diverse different bacteriophage, which has been shown, in different kind of study, has a very important role in determine the structure of the bacterial community. And also, there are recently identified what we call the CPR, stand for candidate phylum radiation. So these are actually a group of very distinct, unique, like nanobacteria with a very reduced small size and very unique lifestyle. So recent study has demonstrated that crosstalk between this different group of organisms have a huge impact on the microbial physiology and pathogenesis, as well as microbial host interaction. So, for example, the cross-kingdom interaction between the fungi species, for example, the Candida albicans, and with some of the oral bacterial species, for example, the Streptococcus aureus and Streptococcus mutant and the Fusobacterium nucleatum, they actually can lead to synergistic action during the microbial host interaction and has a huge impact on the disease initiation and progression. So here is the recently identified group bacteria, which I just mentioned, called CPR, the candidate phylum radiation. And it has been recently isolated and also characterized from the oral cavity by our group. And these nano-sized bacteria display a very distinct lifestyle. So as you can see, these are nano-sized bacteria, usually between the 200 to 300 nanometers, okay, with a very much reduced genome. And they have this, what we call the Epi-Symbiotic Lifestyle. So in order to survive, they have to live on the surface of another host bacteria. So these are some of the images showing you, the tiny bacteria that has this tight physical interaction with this host bacteria. And so these CPR group actually can be found in the human oral cavity and has been associated with some of the disease as well. And they can also be found within other mammal-associated microbiome. And intriguingly, using a mice model, we recently just shown that this episymbiotic nanobacteria can actually suppress the gingival inflammation and bone loss in the mice model through the host bacterial modulation. So its role in the health and disease is warranted further, investigation. So what I present you is just a very limited example of interaction between different group of microorganisms and their impact on the host microbial interaction. For example, we have this huge diverse of the conventional bacteria. And on top of that, you have this, very unique lifestyle, like the episymbiotic bacteria, which are called CPR group. And you have this bacteriophage. And on top of that, you also have this diverse fungi, and how these different group of bacteria interact with each other, as well as how them as a group that interact with the host, and they can have a huge impact on outcome, whether the host maintain a healthy or have disease. So lastly, here are some of the challenge and in the current oral microbiome research. So for example, as I was saying, in the oral cavity, now we have over 700 bacterial species, and we can culture over 70% of them. But what's the rest of 30%? And we still don't know much about them. So one of the goals for us is more like to really culture those yet to be cultured bacteria and for detailed study. And then what's the link between the oral and the systematic disease, and to finding the systemic or have a very very detailed basic understanding of this. And the third is from association to causation. And I think this is a big question, because most of the study right now is still stay at the association level. How we can move forward to the causation research is a very big issue. And then it's to address some very fundamental questions. For example, how these diverse bacteria and microorganisms can get together, how they assemble it, how they have this very dynamic stability and resistant to disturbance. So what's the principle governing these very basic microbial ecological features? And then, as I was saying, it's very important. We need to achieve this holistic view of intro and the cross-kingdom interaction, as well as the microbial and host interaction. And eventually, we want to have this novel and effective solution for us to fight the disease such as the dental caries and the periodontal disease. So these are some of the challenges, but at the same time, this is also an opportunity. So it's my hope that by taking advantage of the new tool, new technology, and also with the holistic mindset, we will achieve a better understanding of the oral microbiome, and hopefully we can decoding the key to a better oral health. So I want to thank all the research team whose work has been cited in my talk, and also of course those who work that are not cited because of the time constraint. And thank you all.
Doctor, thank you very much for your very comprehensive talk from the history of microbiology to the very advanced, technology and the knowledge about microbiology. I'm very impressed. Thank you very much.
Thank you.
And now it's open for the discussion, and but we don't have any question or comment in the chat. But so, do you have any question from the speakers? May I? Oh,
yes, I
yes.
can maybe ask something. So
yes.
Thanks very much for your talk. I just wanted to know a little bit more about the CPR and especially TM7. Could you give us any insight into their general role and how about their lifestyle, things like this that might be of interest to people?
Yeah, so this is the first a CPR group. Actually, if you look at the literature right now, actually CPR group is more and more metagenomic data coming out. So indicating, CPR group is not just can be found in the oral cavity. They're actually in the environment, a huge, diverse, different CPR. So by estimation, it actually accounts for over 25% of the microbial diversity. So there's a huge amount out there. So our lab actually isolate the first CPR. And in the past, the reason people cannot isolate them is because of the lifestyle, because you cannot grow them as a single colony, because they cannot grow by themselves. In order to grow, they have to have this tight association. They need a host, a bacterial host. So they have this what we call epi-symbiotic lifestyle. So they grow on the surface of other bacteria. And if you look at the genome, they have a very much reduced genome. So they cannot produce their own amino acid. Some of the membrane compound they cannot produce by themselves. So they have to rely on the host. And so also very interesting is that if you look at literature, by doing the sequencing, actually the TM7 relative abundance increased in the periodontal patient. So the old thinking is always, hey, you have a higher increased abundance, so it has to play a very active role in the periodontal disease, right? So actually, the recent study which was published in the host cell and microbe is that actually it's not like what we thought. So it actually can have this modulating effect on its bacterial host. Actually, have a somewhat what we think it might be the protective effect. Yeah, the reason is grow well is because they love those, the nutrients when you have a periodontal disease, and they just love those nutrients. And so in the case, usually in one host bacteria, you can have like multiple 3 or 4 different TM7 associated. But when you have this nutrient coming from the periodontal pocket, you actually, for every one of those host cell, you can have 50 TM7 associated. So that's just says to the relative abundance, so it's very fascinating. We're still trying to figure out more, in terms of interaction.
Okay,
yeah.
Thank you very much. It's very interesting, but due to the time limitation, so maybe we have to go to the next speaker. Thank you very much, Professor Dr.
Thank you.
Okay, thank you very much. So now I would like to move on to the next speaker, Dr. Junpei Washio from Tohoku University Graduate School of Dentistry. He is currently the junior associate professor, actually in my lab. So his research focuses on the metabolism of bacteria and microbiome. So I think his talk today will build on his impressive series of work. So please start your presentation.
Thank you very much. Committee. I already prepared the movie.
We received a pre-recorded video. So could you start a video?
Okay. Hello everyone. I'm Junpei Washio from Tohoku University Graduate School of Dentistry in Japan. Before I start, I would like to thank the organizing committee of this symposium for inviting me. Today, I would like to talk about metabolic functions of the oral microbiome. How does the oral microbiome function and contribute to our health? My presentation is in these 3 sections. Firstly, I will talk about section 1. In oral microbiome, huge numbers of bacteria are living and creating their community. Because these bacteria are alive, they have to obtain energy for survival. For that, they metabolize substrates like sugars and amino acids with the production of various end products and intermediates. These end products are very important because they relate directly to the onset and progress of oral diseases. For example, the organic acid produced by bacterial sugar metabolism causes dental caries, while short-chain fatty acids, ammonia, sulfur compounds produced by bacterial protein and amino acids metabolism cause periodontitis and oral malodor. Thus, the more these metabolites are produced, the more risk of these diseases may also increase. Therefore, metabolic function is an important key for the etiology of oral diseases. Recently, the metabolism of the other substrate is also attracting attention, especially the metabolism of ethanol and nitrate. Oral bacteria can produce acetaldehyde from ethanol and nitrite from nitrate, respectively. Acetaldehyde is known to have high carcinogenicity. Therefore, acetaldehyde might contribute to the induction of oral and upper digestive tract cancer by exposure to oral tissues. On the other hand, nitrite is known to have antimicrobial effect and vasodilator effect. These effects may contribute to prevent oral diseases and systemic cardiovascular diseases and maintain our overall health. This view points that the metabolic functions of oral microbiome has a positive role in our systemic health is very attractive to us. So far, many bacteriological studies on the oral microbiome have been performed. Most of them are about who are they, that is, the search of the composition of bacteria in the oral microbiome, or the number or proportion of bacteria that are considered specific to oral diseases. These information are very important, but even if the targeted bacteria are present, they would not have any effect unless they are metabolically active. For considering the true pathogenicity of the oral microbiome, not only who are they, but also what and how are they doing is important. That is, clarifying what kind of metabolites are produced and evaluating how its producing activity is important. Furthermore, bacterial metabolism can be modified by various environmental factors in the oral cavity. Through investigating these effects on their metabolism, estimating its regulatory mechanisms is also important. So, how can we analyze the metabolic functions? Roughly divided, there are two types of the method. One is to monitor the metabolic activity in real time. The other is to measure the amount of metabolites produced. In order to investigate the metabolic functions of the oral microbiome, we have conducted various metabolic studies using the representative oral bacteria or the actual oral microbiome as samples. Firstly, I will introduce pH-stat system. This is a pathway map of glucose metabolism. When oral bacteria metabolize glucose, various acidic metabolites are produced. This system can monitor the acid-producing activity, that is, the speed of its production in real time. pH-stat system consists of the base unit, pH sensor, and titration nozzle of alkali solution. The reaction mixture contains the bacterial cells, buffer, and substrates such as glucose. When the pH in the reaction mixture is lowered by bacterial acid production, alkali solution is automatically added to keep the setting pH for the nozzle. The pH-stat system can monitor the amount of alkali solution added continuously over time. The amount of alkali solution corresponds to that of acid produced by the bacterial cells. Like this, we can monitor the acid-producing activity in real time with this system. In our laboratory, this system has been applied to various metabolic studies. However, we could evaluate only acid-producing activity and not applicable to very small volume of samples such as the oral microbiome. As another way to estimate metabolic activities, I will introduce fluorescent dye system. Please see this pathway map. In the living cells, the reducing power such as NADH, NADPH, and so on was always produced through various metabolic pathways. These reducing power can be monitored using fluorescent dye and fluorescent spectral photometer. Therefore, we have tried to use this reducing power as an indicator of the metabolic activity. As a fluorescent dye, we used Arama blue. Arama blue contains non-fluorescent reagent resazolin. This resazolin is reduced easily by reduction power produced by bacteria and changed to the fluorescent reagent resolvin. By monitoring the increase of the resolvin using fluorescence spectrophotometer, we can evaluate the metabolic activity. The advantage of this system is that it can evaluate the metabolic activity derived from various kinds of substrates, even in relatively small volume of samples, such as the oral microbiome. For quantitative measurement of metabolites, these methods have been often used. These methods helped us to understand the relationship between specific metabolites and the pathogenicity of oral diseases. However, Most of these methods can only measure a limited metabolite. Ideally, in order to understand the full scope of metabolic function in oral microbiome, it is desirable to analyze various kinds of metabolites simultaneously and comprehensively. Furthermore, most of these methods need relatively much volume of sample. Therefore, the analysis of metabolic property in actual oral microbiome has technical limitation. Actually, in these 2 decades, the techniques and devices for metabolomics have been developed rapidly, which has helped to solve these technical limitations. One of them is CE-mass, consisting of capillary electrophoresis and the time of flight mass spectrometer. This device can identify and quantify almost all ionic metabolites in the cell comprehensively and simultaneously, even in very small amount of the samples, such as the actual oral microbiome. When comprehensive analysis of metabolites are available, metabolic activities can be estimated by reconstructing metabolic pathways, which greatly contribute to clarify the metabolic properties in actual oral microbiome. Then, as section 3, I would like to share some of the results of our recent metabolic researches. Firstly, I will introduce the research on ethanol metabolism. I will show the result on the effect of environmental pH, oxygen concentration, on ethanol metabolism by oral Neisseria and Streptococcus species. These bacteria are known as indigenous bacteria and have been detected as an acetaldehyde-producing bacteria in previous study. The bacterial suspension of these bacteria was incubated with the buffer for pH adjustment and ethanol as a substrate for 30 minutes in aerobic or anaerobic conditions. And the acetaldehyde production is quantified with sensor gas chromatography. The upper graphs are in aerobic, and lower ones are in anaerobic. The horizontal axis shows the pH conditions, and the vertical axis shows the amount of acetaldehyde produced. The amount of acetaldehyde produced varied greatly depending on the pH and oxygen concentration conditions, and the behavior of the changes varied depending on the bacterial species. Thus, the actual metabolism of ethanol by these oral bacteria was greatly affected by various environmental factors in the oral cavity. Next, I will introduce the research on nitrate metabolism. In our previous studies, we have shown that these bacterial species are the main nitrate-reducing bacteria in the oral cavity. In particular, Actinomyces, Scharier, and Venerea species have been detected in large numbers. These bacteria are also known well as indigenous bacteria. Here, I will show the results of nitrate metabolic activity by oral Venerea species and the effect of environmental factors. Venerea atypica is cultured in the medium with or without nitrate and nitrite, and the bacterial suspension was prepared. The suspension was incubated with the buffer for pH adjustment and nitrate as a substrate, and lactate in anaerobic and anaerobic conditions. And nitrite production is measured with grease reagent. I will show the result. The horizontal axis in each graph shows the lactate concentration, and the vertical axis shows the amount of nitrite produced. Gray bar is the production by the bacteria grown without nitrate or nitrite. Green bar is with nitrate, and orange bar is with nitrite. As the lactate concentration increases, the amount of nitrite produced also increases greatly. In further, environmental pH, nitrate, and nitrite also affected greatly on the nitrite producing activity of this bacteria. These results suggested that the metabolism of nitrate is also greatly affected by various environmental factors in the oral cavity. As explained before, Bayonella species is numerically behind Actinomyces and Charia among the nitrate-reducing bacteria in the oral cavity. However, even if the number is less, oral Bayonella may actually account for the most of the nitrite production in oral cavity, depending on the oral environment. Thus, it is important to consider not only the kind and the number of bacteria, but also their metabolic functions. Next, I will introduce the metabolomics of actual oral microbiome. We tried metabolomics of supra-gingival oral microbiome samples before and after glucose rinsing in vivo. We reported a lot of results of sugar metabolism by oral bacteria, but this is a world-first study on the metabolomics of oral microbiome. As an example of data, I will show the results of the metabolic intermediates in the EMP pathway. As shown in this graph, various metabolic intermediates were detected, even in very small amount of clinical samples, and the increase or decrease of these metabolic intermediates could be clearly detected. This figure shows the major pathways involved in glucose metabolism. In the figure, red letters show the metabolites increased after glucose rinse, while blue letters show decrease. The fluctuation of all metabolites levels suggested that these pathways actually functions in the oral microbiome. Next, this graph shows the fluctuation of amino acids level with glucose rinsing. Only alanine, glutamate, and cysteine increased, while the other all amino acids decreased. These results suggested that when cells metabolize glucose, most amino acids were consumed. Thus, we have shown that metabolome analysis using the actual oral microbiome is possible. So, as next trial, we conducted the metabolomics research about the effect of xylitol and fluoride on oral microbiome. Xylitol and fluoride are known well to inhibit the acid production by oral bacteria, but the detail of effectiveness was confirmed in the experiment using cultured bacteria in vitro. Then, we collected the supra-gingival microbiome samples as shown in this slide. At first, I will show the effect of fluoride in vivo. This graph shows the proportion of each metabolite when the fluoride rinse was applied before glucose rinsing in the metabolites after rinsing with only glucose. The amount of each metabolite increased or decreased by fluoride, suggesting that some effect by fluoride occur in the oral microbiome in vivo. Similarly, this graph shows the proportion of each metabolite after glucose plus xylitol rinse in the metabolites after only glucose rinse. The amount of each metabolite was not changed by xylitol. These results showed in the oral microbiome in vivo, the inhibition of acid production by xylitol was not observed. Most knowledge about metabolic mechanism in the oral microbiome available today is not based on in vivo data. Instead, it has been estimated from the in vitro data using cultured bacterial cells of the representative oral bacteria. Of course, Conventional in vitro metabolic studies using representative bacteria were equally important to determine and confound the metabolic network in the oral microbiome. However, we have to notice the metabolomics using actual oral microbiome is essential for the development of the accurate diagnostics, effective treatment, and preventive strategies for oral microbiome in future. I would like to conclude this presentation. Firstly, metabolic functions of oral bacteria in the oral microbiome contribute to both oral and systemic health. Hence, the study on the metabolic functions is important for oral microbiome research. Secondly, the analysis of metabolic functions using not cultured bacteria but actual oral microbiome is very essential. The whole pictures of metabolic system and network in the oral microbiome is still unclear. Further challenge is needed to pioneer new technologies and gain a deeper understanding of oral microbiome. I would like to acknowledge to all members shown in this slide. Thank you for your kind attention.
Thank you very much, Dr. Washio, for your comprehensive study about microbiome metabolism. I think it's also the kind of the microbiome study from the different point of view using metabolism. Thank you very much. So now it's open for the discussion. Actually, I already get one question from the audience. Oh, it's Dr. What, maybe? Which species of Actinomyces are you studying? Are there big difference between different species and strains?
Thank you very much. We used our Actinomyces Nesrandi, maybe ATCC 12104 only in that study. But at that time, we compared the metabolism of the Actinomyces with the metabolism of the other bacterial species, such as the Streptococcus species. So we don't have enough data about the other Actinomyces species, but we have other species of Actinomyces and the Sharia Monolyticus now. So in future, I will try to do yes. Thank you very much.
Thank you. Yeah, you're right. There are, various Actinomyces is different. And also the naming is very complicated. During history, it's changing each time. So.
Exactly. So it's really a pain. Actually, we recently just submitted a paper and talk about the actino, and then we realized, hey, there's a new.
Yeah, that's right, Sharia or something like that.
Yeah.
That's right.
Yeah.
Thank you for your good comments. Yes.
Thank you very much.
And now, so time is limited, so I'd like to move to the next speaker, Bart. So next speaker, Dr. Jaya, is absent, maybe from the emergent matter. So, so I'd like to skip him and move to the last speaker, Dr. Watt's presentation. So if he come back, Dr. Yaja will come back here, he can present his research, but if not, we'll have a little bit big discussion time. Okay, so now we move to the next speaker of the Dr. Watt. So could you please start the pre-record video for him?
Hello, my name is Rory Watt. I'm from the Faculty of Dentistry at the University of Hong Kong, and the title for my talk today is The Twists and Turns of Periodontal Disease, Etiological Insights and a Novel Treatment Approach. So there'll be two main parts to my talk today. The first part will be about oral treponeme bacteria, which has been the subject of my investigations for several years now. And the second part will be about using a microbiome transplant to try and treat periodontitis in a canine model. So if we think about the oral microbiome in health and disease, we think about two main states, eubiosis, the healthy state, and dysbiosis, the disease state. So this refers to the combined community activities of the oral microbiota present within the particular niche. We have host, environment, diet, oral hygiene factors may all play a role in changing the composition of the oral microbiome. In caries, we generally have a species are depleted, we have lower diversity, and in periodontal disease, we have higher diversity and it's species rich. But we don't really know how to exactly or precisely define a healthy or a dysbiotic microbiota. We have commensals, keystone pathogens, pathobionts, accessory pathogens, certain community ecotypes, which all may play a certain role. And that might be different within different niches and different hosts. But what we tend to think of nowadays is, as part of the ecological plaque hypothesis, is the term resilience, the resilience of the microbiome community to resist change and to retain its state. So for most people, that would be the healthy state, a robust eubiotic system. But in disease, we may go towards a metastable, semi-dysbiotic state. And then if pushed further, this can go to a firmly established dysbiotic state. And that's after this certain threshold. So we must, if we're thinking about treatment, we must either prevent the change or take action before it or at this metastable dysbiotic state. So as I mentioned at the start, I've been investigating treponines for several years now. So these are quite important for human and animal periodontal infections and other polymicrobial infections in animals, tissue-destructive diseases. In the oral cavity, Treponema denticola has a number of well-established virulence factors, and it's associated with periodontal and peri-implant disease. But the ecological and etiological roles of other treponemes are far less clear, and we need improved high-quality genomic information so that we can identify these and classify these more precisely, and that will facilitate downstream metagenomic analysis. Also, if we're looking at the oral cavity, we want to look, are there certain lineages of these species that might be more prevalent or more pathogenic? For oral treponemes, we have 50 to 70 species or species level phylotypes in the oral cavity. And the taxonomic system places these within 10 phylogroups, which may contain one or more species or phylotypes. So I'm most interested in phylogroup 1 and phylogroup 2 here. And so phylogroup 1 contains T. vincentii, T. medium, and many T. vincentii, T. medium like taxa. In phylogroup 2, we have Treponema denticola and another species that is not commonly discussed or investigated, Treponema putidum, and that appears to be possibly more relevant for animal infectious diseases, but we find it in humans. So in this first investigation a few years ago, my group investigated sequenced 4 genes within 72 human oral treponeme isolates that covered at least 6 or 7 species level phylotypes or species. And these were obtained isolated in 6 countries spanning 3 continents. So the analysis of these genes quite strongly supported the existence of 3 completely separate species level phylotypes spanning between T. medium and T. vincentii. And these were called T species 1A or Treponema sinensis, that was proposed by the original isolator, Dr. Chris Weiss, and Treponema species 1B and Treponema species 1C. So after that initial MLSA analysis, we look at the whole genomes. So here's the phylogroup 2, so Treponema denticola, Treponema putidum. So we find they're well separated, but we have these two atypical T. denticola strains here. And these really are on the borderline between new species or subspecies, something like that. So 23 strains analyzed. We have a pangenome of over 10,000 genes, about 500 core genes, an accessory genome, and then the non-treponema genetic origin genes come predominantly from bacilli, Clostridia, and Bacteroides. If you look at the phylogroup 1, treponemes, if we look at the whole genome data, this supports, strongly supports the existence of these 5 species level phylotypes, A, B, and C. And so the functional analysis of these genomes is ongoing at the moment. So this is unpublished work I'm talking about at the moment. So Treponema species 1b and 1c are commonly isolated in animal niches, but are also appear to be quite prevalent in the human oral cavity as well. So there are maybe 8 to 12 established Treponema denticola virulence factors. The most important of these are probably the dental-lysin protease complex and the major surface protein, but we have several others. And so the various taxa that I described, the T. putidum, T. denticola, T. medium, T. vincentii, and the other 3 have various sequence types and various there are sometimes absent, sometimes present, sometimes quite different forms of these genes. And so this is probably the basis of their differing ability to cause periodontal disease. So they're not, none of the other species seem to have the same complement of these virulence factors that Treponema denticola has, especially the dental-lysin protease complex appears to be particularly different across these species, and I think that's playing a particularly important role. So this is the subject of current investigation. So if we've looked at the genomes, the genomic information, I'd like to apply that information to look at their distributions in clinical settings. So here, this is a small scale study, a sort of focused in-depth look at mild to moderate gingivitis, 10 subjects, and more severe forms of generalized localized periodontitis. So we're wanting to look at these Treponema vincentii, T. medium, and the T. denticola-like taxa in these disease niches. So a focused look. So we do this via analyzing just one gene. So our analysis indicated that the PIRH housekeeping gene is an excellent marker of diversity and taxonomy, very strong correlation, and we're able to discriminate different species with high confidence. So we just analyzed the sequence of this gene in depth in all the subjects, and then we can identify the Treponema lineages present. So there's a lot of information on this slide, but the take-home message is the approach is very specific. Every single gene we obtained was PIRH and very broad. We covered at least 7 or 8 species level phylotypes, many novel taxa identified, and we classified these within clinical PIRH genotypes, which is more detailed than the species level. So it's a good way of getting some high resolution assessment of the different Treponema species or phylotypes that may be present in these disease niches. So the interesting thing to note, or probably the most interesting thing, was that individuals harbor multiple completely different lineages of the same species. So most notably Treponema denticola. So some individuals may be infected with 5, 6 treponema, probably 5 or 6 Treponema denticola, at least 3 with high confidence, because there's still genomic space we've not investigated. One lineage, which was highly related to the type strain of T. denticola, was highly prevalent in 16 out of 18 subjects. One lineage was highly prevalent in periodontitis, and 2 lineages were associated with mild to moderate gingivitis. So this highlights the more nuanced relationship between these types of treponemes and periodontal health status. It's a quite a complex picture. So the second part of my talk is about a canine oral microbiome transplant. So we're trying this as a analogy with the fecal microbiome transplant, which are used for the treatment of chronic inflammatory bowel infections, such as that caused by C. difficile. So we decided to use a canine model because of size, and the etiology and microbiome of dogs is very similar to that of cats and humans. And also age-associated periodontal disease is very common in canines. So by the age of 2, more than 3/4 of all dogs will have periodontitis or of some level of severity. And so we had 18 systemically healthy beagle dogs with age-related periodontitis. These were randomly assigned to 2 groups, the recipients of the microbiome transplant or controls, and a single healthy, systemically and periodontally healthy 4-year-old beagle dog donor. And this was led by Professor Thomas Beichler, then at University of Düsseldorf. And this received full ethical and animal safety oversight. So here we have a video of the procedure, the donation procedure. So the microbiome transplant is resuspended in reduced transport medium and then applied over as many surfaces in the oral cavity, focusing on the gingival margin of the dogs that are under a general anesthesia. So the time point of the process I'll show in the next slide. Here we are, it's a little bit complicated, but basically we have 4 time points. We have the pre-baseline, the week -2. So here we perform a clinical examination, collect samples, do a full mouth debridement to reduce as much of the original microbiota as possible. 2 weeks later, after there's some establishment, we do the clinical exam. We take microbiome samples. We do the full mouth debridement. Then we give the transplantation or not for the controls. 2 weeks later, we do the clinical exam and do the microbiome analysis. And then 10 weeks after that, we take samples and do a clinical exam. So it's a very complicated procedure because we use a single donor. So the single donor dog has to be used multiple times using a split mouth design. So it took 18 months to actually conduct the trial. If we look at the taxa that we find within the donor, control, and recipient dogs, we see it's quite typical of canine, dominated by Bacteroidetes, Proteobacteria, Firmicutes, lesser levels of Fusobacteria, and then slightly less Spirochetes. And so this is just typical Illumina-MiSeq amplicon sequencing, assigning taxa using the Data2 pipeline into amplicon sequence variants, which are like an OTU. So if we first look at the alpha diversity in the dogs across the groups, we can see that both the recipients and the control dogs have a higher alpha diversity than the donor dogs. So this reflects the disease status of these recipient and control dogs compared to the healthy donor. So all of the dogs have a decrease in alpha diversity after the baseline. So this just reflects a better health status after removing the microbiota. But the main difference here is in the baseline to week 2. So what we find is in the microbiome recipients, we find that between the baseline, where they get the transplant, and week 2, there's a further reduction in the alpha diversity, consistent with our improved health status. But in the controls, this rebounds and goes back, and then rebounds again to the final time point. In both sets of dogs, they rebound to a sort of similar level over the time course. But it's this very short-term effect that the evidence suggests this is a reduced dysbiosis during this time point. So if we look at the beta diversity in these dogs, we see that at the week -2, the baseline, and week 12, the donor and recipient dogs are very similar. But at this week 2, where we believe there's the near maximal effect of the oral microbiome transplant, we see a notable shift of the recipient dogs, and there is the maximum overlap or the closest relationship between the recipients and the donor dog. So in this animation, we see the 3 time points, so we can see the conserved trend. If you compare the movement of the controls and the movement of the recipients, you'll see this conserved 3-point shift. So it's a much more, the change is much more consistent across the different recipient dogs. And we believe this is induced by the changes. The changes are induced by the introduction of these taxa from the oral microbiome transplant. So also, we can do further analysis to try and show the effect of the transplant. So we can identify 70 ASVs that are transferred from the donor to the recipients. They were not there before. So that's strong evidence of the at least transient colonization by these supposedly healthy taxa. We can also statistically look at the changes in the microbiome composition using the PRC curve analysis. We can look at taxa that are decreased or increased. And interestingly, we find Porphyromonas gulae, which is the equivalent to Porphyromonas gingivalis in humans, has decreased in the recipients compared to the control dogs. So what conclusions can we draw about this? So to the best of our knowledge, this is the first ever oral microbiome transplant to treat some form of disease, periodontal oral disease, periodontal disease here. It was a long and technically challenging study with no precedent, so we made some decisions that ultimately may have been, we could have used an alternative approach, so we learned many lessons, positive and negative. I would say that if this were to be repeated, we would need multiple transplants of the donor. We'd need increased biomass. We need more plaque going into these recipient dogs. And possibly we might want a matching of the donor to the recipients. So this is analogous to the fecal microbiome transplants, where we have so-called super donors that are super effective at transplanting bacterial taxa to hosts. So an Australian group have recently been described something like this, still in a mouse model, to work out the procedures that they need to do this in humans, which will be a lot more challenging for various ethical, safety, and such reasons. But I believe this will work at some point in the future. I think it will probably be at least 10 years until we get some sort of established protocol. But that may be something that we can look forward to. So I'd like to thank my colleagues and collaborators on this project, especially Professor Tom Beikler and Thomas Fleming, the other workers in Hamburg, and my colleagues and collaborators here in the University of Hong Kong. And thank you very much for your attention. Thank you.
Thank you very much, Dr. Watt. Your very interesting presentation about diversity within the treponema species, and also the very interesting trial, the canine oral micro transplant. It is very interesting. Thank you very much. So the presentation is now open for the discussion. Actually, I have a question for you from the audience. So,
thank you.
Okay, where are treponemas mainly found in the oral cavity?
So they're being anaerobic. You'll always find them predominantly in subgingival niches, but healthy and diseased, I think, have very diverse populations there. We all have them, but the proportions may be very different, and the balance of the different species may be very different. But we'll find them on the tongue, tonsil crypts, many different other niches as well.
Okay, and also there is a big difference between healthy and healthy people and healthy periodontal disease? It's also the question from the audience.
So yes, so I think the thing is obviously I think many of the panelists have written here about it. I think it's what they're doing as opposed to what is there. So it's the combined activities of the treponeme species, and they were working in consortia with other taxa. Porphyromonas gingivalis is known, I think. Fusobacterium, probably there'll be other taxa as well. So I think it's the whole regime shift. The community changes over from a healthy state to a more diseased state, and they're playing a role, and it's hard to tell what is direct and what is indirect.
Okay, and the other question from the audience: what are actual pathogenicity differences between species? Yeah, of treponema. This is also the question for the treponema.
So yeah, so I think the thing with the treponemes is I think many of them are actually pro-healthy. So we tend to think of them as being promoting disease, but I think many of them are actually playing a sort of protective role. They occupy the niche to prevent the more disease promoting one from taking over. I think there's a balance between many of them, but that I can't prove that.
Okay. Also another question for the treponema. So do you know some physical relationship of the treponema with P. gingivalis?
So there's metabolic symbiosis and physical. There is so that's a strange one, because obviously treponemes are very motile, but I think it's a lifestyle factor. There's papers where they have P. gingivalis hitching a ride on the Treponema denticola, like riding a train. But I think it'll be very much dependent on where they are and what they're with and which tissues, which type of biofilm. I think it's not a straightforward question.
Okay.
Or not a straightforward answer.
That's a good answer. Thank you. And now we'd like to move to the next speaker, but Dr. Jaya Seneville Latne. He's going to be the absent, maybe due to urgent matter. So today we'll skip his presentation. So we're going to the discussion time now. Is okay? And actually, first of all, thank you very much for your very comprehensive and great presentation from the three speakers. We learned a lot about microbiome study. And okay, now first, do you have any questions or comment to each other?
Yeah, so actually,
please.
So Roy, very nice talk, very nice talk. Actually, I have a couple of questions for you.
Okay.
So for the so for the TD, right? So a while ago, actually, I read a paper about brain microbiome. And essentially, also, recently there's this link of the oral bacteria with some of the brain disease, like Alzheimer's disease, right? So recent paper about the PG, how the potential role. So I'm thinking about the TD, because as you were saying, TD really motile, right?
Yes.
So in order for bacteria, because I'm not sure, now I still don't see any kind of concrete evidence to say these bacteria actually can, get into the brain, because as you can imagine, the brain, the blood-brain barrier, and it's going to be very difficult for regular bacteria to penetrate and get in there. So, but considering the TD, right? It's really motile, and what's your thought on its potential link, or do you know any kind of evidence suggesting they can be there, or any kind of metabolite derived from the TD and is associated with the potential, the brain, some of the brain disease.
Right. So no, thanks for the question. So I think I do remember seeing some papers on this, but I can't. It's a while ago since I read those. I can't remember the specific argument. So Treponema pallidum obviously is known as being an invasive species, and you can get neurosyphilis. They can get nerve infection. Or central nervous system infection, but Treponema denticola, I think it's one of these ones that it's it could be what you're seeing is, well, I don't particularly think it can go that far. I think it can be maybe invasive tissue infection, and maybe obviously there could be some molecules released. So I think the thing with T. denticola is maybe the key thing is the outer membrane vesicles. So similar to P. gingivalis, it's shedding these outer membrane vesicles. They can go into the cells. They can penetrate potentially anything. They're micelles. They should be able to transfer widely. And so there could be genetic material in there. There could be RNA. There could be, proteases, a bunch of immunogens. We're not sure exactly. I mean, there's some studies on that. So I think the effects from that could be related to that. But I'm not convinced about the PG and the neurological conditions, but I could be wrong. I could be wrong.
Okay. Thank you. Yeah, the second question is about the transplant. I really like the study you did. Yeah. So as you were saying, this is probably the first trial, right? Attempt to do this. So based on the result you see, right? So compare with the control and your transplant group at the week 2 after your transplantation, you definitely see a kind of some kind of indication as you see the reduced diversity of the bacteria, which is the indication. So, but after like a couple of weeks, it's kind of go up. So, what's your thought on this? Is it because of those bacteria? Because it is one single dose, right? Is it single dose?
Yes.
It's a single dose, and that's probably making them really hard to stay. But because as they can imagine, your saliva constantly swallowing, and really is a challenge for the introduced microbiome try to kind of establish them. So, do you think instead of one shot and maybe every other day, and maybe give a few more this type of treatment, and could potentially make the situation better?
I think almost certainly that's true. So I think what we saw is basically there was many, because of the cost and logistics, it was basically a one-shot experiment. We just tried to do it, and then that's with many restrictions, and the working with and so the amount of plaque we can put in is small, and the mouth is large, and it's transient, and so what you say is almost you must do that. The question is matching donor to recipient. I think will be very, very important. The pre-treatment, the cleaning, obviously with gut, you flush out as much bacteria, antibiotic and flushing. Oral cavity, that's more difficult, but you can do obviously very deep cleaning of all oral surfaces. The tongue will be a big reservoir, I think. And so you want to remove as much indigenous plaque and then transfer as much of the donor plaque in, and then do that repeatedly. And the thing is, how much, how often, do you put it in some cream formulation? Do you want some like a slow release, or I don't know. So and it has to, if you're talking periodontal, as it penetrates. I mean, that's a physical barrier. So you have to, open. It's very complicated. But I think in theory it should work. It's just going to be technically difficult, and there's many things that need to be worked out, I think.
At the same time, you said that the super donor, super donor word. So what kind of microbiome should be used for the transplant?
I think you would have a better idea than I would, I think. You need to do a big study of, so I think this is what the Australian, I mentioned there's a paper from some Australian researchers who I think are doing this. They're trying to work on this problem, thinking of the super donor concept, and then also propagating, in vitro propagation, growing the bacteria. So obviously you're changing the composition, but hopefully you'll retain enough of the pro-healthy species that you can then transplant them repeatedly into the recipient. So that's the thing. We don't really know, because it's, it's like looking at nature. Is that a healthy forest? Is that a healthy forest? Is that a healthy field? It's all different. So I think that's
just like what you say, what is how to define dysbiosis, right? Because
yes.
in a way, it's really hard to define what is eubiosis, because different,
Yes.
inter-individual like a difference, and so that's, it's very diverse. It's very diverse.
Excuse me, I had a question from the audience to the Dr. Watt again about the treponema. It is relatively difficult to grow the treponema in a usual medium. So, do you have any idea to grow the treponema and isolate them?
So I have to admit that there are many people in the world much better at cultivating treponemas than I am. And so many of them are in Japan. So I don't know if this is coming from someone who knows somebody. I think there's some very good oral spirochete researchers. I think the key thing is stability of your anaerobic chamber. Some of them are very sensitive to oxygen fluctuations. So you need to have a chamber that people are not opening and closing all the time. There's all these people doing PG experiments. They're just taking things in and out. Oxygen's going up and down like this. They need a lot of, so I think not as bad as Tannerella. I think they're very sensitive. Treponemes, definitely. And then I think there's a lot of difference between serum. A lot of them like serum. So I think that can be a key factor as well. And maybe just batch to batch variation. And yeah, I think there's a lot of factors, and they like a high density. So starting them is always very difficult. They need to once they get to a certain density, then they seem to be a lot more robust.
Okay.
There are many people who can, I can refer the person to speak to who can give much better advice.
Okay. Okay, now
I still have a question for the.
Okay,
for
okay, please.
Sorry, it's
That's
about
okay.
the TD.
Still, we have 10 minutes. Don't worry about it.
Yeah, so because back at the UCLA, actually, one of my colleagues, he's also working on the treponema, the TD. So I'm curious, and so how frequent you can detect the TD from, let's say, healthy subject, with no periodontal disease, and so what's the recovery rate you can have?
It's there. It's always there if you look hard enough, but the level is low. But they're indigenous. I think the question is what I was trying to look at but not managed to answer is just actually, all these very strange anaerobics, anaerobes that we get. Obviously, we are colonized by them at some time from caregiver, from parent and stuff. And obviously our dentition changes, but they're in there and they're coming from wherever, but they're all there. We're not developing the disease because they've come in and then they take over. They're already in there in the healthy plaque. And then there's this balance between all the different species, but you will find T. denticola basically in everyone. You just need to look, do enough sequencing depth, and you will find it, I think.
Okay.
Thank you. Yes.
So can I have a question to the Dr. He?
Yes, I would like to ask one. I'm interested in your bacteriophage. You mentioned the surveying bacteriophage, or that area for the oral microbiome, and then the whole pangenome. Could you tell a little bit more about that?
Right.
Yeah. So for the human oral microbiome database, which is established by the foresight and still maintained by foresight, and now they have this extended version. So essentially, they're gonna include the bacteriophage that can be found in the oral cavity, as well as those microbiome, basically the fungi species. So for the bacteriophage, and we all know, if you look at the literature, all those environmental study, right? The bacteriophage actually play a very crucial role in terms of, inducing this very dynamic structure change, functional change of those environmental microbiomes, like a community. So in the oral cavity, actually, there's estimation, the number of the phage is like 30 times more than the number of the bacteria. So essentially, there has to play a role. And actually, our lab also interested in this. And what we found is interesting that so if you use a bacteria, and then you just kind of to screen the saliva, and you can always find a phage for a type of bacteria you're using. Okay, so basically saying, you know, it's always there. And however, because of some of the, you know, technical like a difficulty, and in terms of to understand their the bacteriophage role in terms of you know, shaping this oral microbiome, I think that's still, you know, we thought it should play a role, but in terms of exactly what role, you know, there is no established, well-established like a model system for study of that. I think this is a challenge right now at this point. So that's why you still don't see much of those, you know, the paper talking about the bacteriophage, the role in you know, there is some study starting to coming out in the gut microbiome, and how the gut bacteriophage play a role in terms of shaping, you know, the gut microbiome. But oral microbiome in a way is always lagging behind the gut, right? So, but still people trying to look into it, and they just try to establish the model. Yeah.
Okay, so almost time, but lastly, I'd like to each of the speakers to give a few words about the future research or future planning of your microbiome study. So first, you, Dr. He.
Okay.
Yeah, kind of
so
a take-home
message.
Yeah.
So again,
Yes.
Thank you for the invitation, and it's really a great pleasure to share some of the, you know, recent development, as well as I would say, you know, some of the challenge. So for example, in our lab, we basically trying to use what we call multi-level, you know, system to study the oral microbiome. So from the single species level, which is, you know, basically you still use the reduction model, right? So, but I think this is very key. Reduction allow you to really study the physiology, some of the basic, for example, the basic gene like a function, structure function, and that's the very basic. This is almost the unit, right? You need to kind of to expand, and then this interspecies, because bacteria we know is not always by itself; it always constantly have interaction, and then the multi-species community. So this is more like a develop certain like a model trying to kind of recapture the in vivo like a condition, allow you to do a better understanding. I think lastly, is not the least, is microbial and host.
Right.
What's happening in the interface, because that's really where it counts, right? Because you cannot study microbiome for the microbiome's sake, because it's always, you know, you have to put it in the contact with the host. So that's where it matters. So that's, you know, study how the microbial and host interact, how the host respond to the bacteria. I think that's very important.
Okay, thank you very much. So second is Dr. Washio.
Okay, thank you very much. Through today's symposium, my interest to the oral biome has become stronger and stronger thanks to the two other speakers. And in recent years, various relationships between the good microbiome and overall health have been suggested variously, or similarly, maybe there are hidden, unknown facts in oral microbiome in the relation to the oral and the systemic health, I think. So I would like to clarify the relationship between the oral microbiome and health through our original approaches focused on the metabolic functions. And Dr. He mentioned a little about the uncultured, how to culture uncultured bacteria. Yeah. How to culture, yes. Yes, I need now I need a cultured bacteria in my research. So someday I would like to try to the metabolism of the uncultured bacteria.
Yes,
Please give information next time, please.
Okay.
Thank you very much.
Thank you very much. And Dr. Watt, within 1 minute.
Thanks. So just thank you very much for giving me the chance to speak today. So and being with this distinguished bunch of academics. I'm quite old-fashioned in the way I look, and I'd like to look at the molecular side of things. So I think biochemistry, protein. So most of the work I do is actually I haven't talked about today. So I like thinking at the protein and the functional level in at a very reductionist sort of way, if we can get good sets of data on functions of specific proteins very accurately, and then in species accurately, then smarter people who are good with computers can put all this together in the future, and then we can get a better picture. So I'm trying to put detailed little pieces together for smarter, better people to join up and make some grand concept with.
Yes, that's right. Thank you very much. So almost it's time. I have learned a lot. Thank you very much for the great speakers. And I really hope this will be the good beginning of a new collaboration within us. Thank you very much.
Yes, thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Bye.
Thank you, Professor
Bye.
Session
Bye.
and
Thank you
Professor
very
Wang.
much.
Yes.
Bye.
Bye-bye.
Bye. Thank you,
Very good
boss.
to be.
Yeah. Bye.
I'd like to close this session. Thank you very much.
Thank you very much. Next, we will start the keynote session 5, periodontium and the biological interface between periodontal regeneration and periodontal repair. The chairperson of this session is Professor Minoru Wakamori, Tohoku University. Thank you for your time. Professor Wakamori, please.
May I test my PowerPoint projection?
Yes, please.
Oh, thank you. It works.
Ah, 若森先生、始めていいですよ。
もう始まった時間ですか。はい。
もう時間です。一応、進行お願いします。
はい。Thank you for, ladies and gentlemen, thank you for joining us. This is Keynote 5 session. I am a chairperson for this session, Minoru Wakamori from Tohoku University Graduate School of Dentistry. We have 3 speakers. The first and the second papers deal with mechanical force on periodontal ligament cells, and the third paper deal with periodontal regeneration and repair. The first presentation is about the paper entitled WNT5A released from mechanically stimulated periodontal ligament cells, promote neurite elongation and branching in peripheral neurons. This paper will be presented by me. Okay. Before I start, sorry, いいかな。はい。Before I start, I'd like to appreciate the organizers of this symposium. It's my pleasure to present our recent data. Because of time limitation, I'm going to mention about elongation, but not branching of neurites today. As we know, we have many fine sensors in the mouth. For example, pain, taste, hot and cold sensation, and tactile and pressure sensations. Recent advance of molecular biology has revealed molecular basis of these sensations. Today, I'd like to focus on pressure sensation and their physiological roles. The periodontal ligaments are very sensitive to mechanical stimulation, such as occlusal force. You can feel uncomfortable when you bite even one hair. A hair breadth is about 30 micrometer. The sensation of pressure on PDL, periodontal ligament, is perceived by the cerebellum via the trigeminal ganglion and mesencephalic trigeminal nucleus. Explanation of this slide is not necessary for dentists. The periodontal ligament contains fibroblasts and nerve fibers. The periodontal ligament has these functions, including mechanoreception. Free nerve ending and Ruffini corpuscle receive mechanical stress. Periodontal ligament fibroblasts also receive the mechanical stress. However, it's not clear the interaction between peripheral nerves and mechanically stimulated periodontal ligament fibroblasts. These are radiographic images of cat's mandible. The cat first molar is not erupted at 2 months, but begins to erupt at 5 months and is fully erupted at 12 months, at which the cat can chew firmly. The percentage of immunoreactive area for nerve in the periodontal ligament increases when cat can bite. These suggest that mechanical stimulation may influence the nerve, induce the nerve into the periodontal ligament, elongate the nerve fiber, and or promote the branching of nerve fibers in the periodontal ligament. In another report, when one of the incisor teeth of the rat is shaved and occlusal force is not applied, Ruffini corpuscle disappeared, and occlusal force is not, the percentage of immunoreactive area for nerve decrease. It was reported that Ruffini corpuscle are formed again when incisor of rat continue to grow and return to the state where occlusal force is applied. These results suggest that mechanical stimulation is necessary for the formation and maintenance of Ruffini corpuscle and nerve fibers. However, the detailed mechanisms of nerve travel pattern controlled by mechanical stimulation through the periodontal ligament is still unknown. Up to now, many substances that elongate axons are identified. They are classified into 2 major groups. The first group is called as neurotrophic factors, which include NGF, BDNF, NT3, and 4. They bind to the specific receptors, TRAC-A, B, C, and P75NTR. Thereafter, they activate various signaling pathways that are involved in growth and maintenance of axons. The second group, called as axon guidance proteins, include SLIT, nettling, Wnt. Each of these proteins binds to specific receptor to precisely elongate axon towards the target site and form a neural network. There are 2 types of axon guidance proteins, secreted type and membrane-bound type. Both types have either attractive or repulsive force on axons. It is possible that mechanical stimulation directly induce nerve elongation. But in this experiment, we focused on the indirect nerve elongation through soluble factors. We hypothesized that neurotrophic factors and axon guidance proteins would be produced by mechanically stimulated periodontal ligament fibroblasts and would induce nerve elongation in the periodontal ligament. Let me begin with the identification of the secreted factors. In our experiment, we used the RPDL cell line or RPDLA cells as well as we called them, while we established from the mouse molar teeth of 6-week-old Wistar rat. First, we checked the expression of neurotrophic factors and axon guidance proteins in non-stretched RPDA cells using RT-PCR. Many neurotrophic factors and axon guidance proteins are expressed in RPDA cells. All of these factors affect nerve elongation. Some are attractive factors, while the others are repulsive factors. We focus on attractive factors such as NGF, BDNF, NTF4, and Wnt5A. We examined whether mechanical stimulation increased the expression level of 4 factors in RPDA cells. Using one directional cyclic stimulation with 15% at 0.5 Hz, which emulated the physiological occlusion force. In order to follow the production time course of the factors, we quantified messenger RNA levels using real-time PCR. The expression level of Wnt5A in stretched RPDA cells reached a peak at around 10 hours, but those of the neurotrophic factors were not changed. Therefore, we focused on Wnt5A. Let me move on the second part. The signaling pathway contributing to Wnt5A production by mechanical stimulation in RPDL cells. The orientation of cells starts to change at around 10 hours after mechanical stimulation. Because the morphological change depends on integrin, we examine the focal adhesion kinase signaling, FAK signaling, as a mechanosensor in RPDA cells. In general, mechanical stimulation increases the intracellular calcium concentration, and activate various signaling pathways. In order to confirm that the increase in intracellular calcium concentration also involved in the enhancement of Wnt5A messenger RNA level in the periodontal ligament, we conducted a stretch stimulation experiment with BAPTA-AM, which chelate intracellular calcium like EGTA. The enhancement of Wnt5A messenger RNA level by mechanical stimulation was completely suppressed by BAPTA-AM, indicating that intracellular calcium increase is required for enhancement of Wnt5A production by mechanical stimulation. In addition, we tried to examine the downstream signaling cascade. It is reported that mechanical stimulation activates 3 pathways: MEC12, ARC, PI3 kinase, AKT, JNK, Jun signaling pathways. We monitored Wnt5a messenger RNA level and applied specific inhibitor to PDL cells in order to block these signaling pathways. The enhanced level of Wnt5 messenger RNA by mechanical stimulation was inhibited by PI3 kinase inhibitor, LY, MEK1/2 inhibitor, U. On the other hand, this is a JNK inhibitor had no effect on Wnt5 messenger RNA level. MEK1/2 and PI3 kinase pathways are involved in the mechanical stress-mediated Wnt5 expression in RPDA cells. Next, we examine the phosphorylation of ARC and AKT. In Western blotting, both AKT and ARC were phosphorylated in the DIMSO control. After 1 hour mechanical stimulation, but phosphorylation was attenuated by PI3 kinase inhibitor, LY, and MEK1/2 inhibitor, U. In this experiment, we found that the mechanical stimulation phosphorylated AKT and ARC in RPDA cells. Next, we identified mechanosensors. Mechanosensitive channels such as TRP, TRP channels, and adhesion factor systems such as integrin FAK pathway are known to be mechanosensors. First, we used RT-PCR to identify mechanosensitive channels. The expression of TRPV1, V2, V4, TRPC1, 5, C6, and C6 was confirmed. TRPV family broad inhibitor, ruthenium lead, attenuates the messenger RNA level of Wnt5a. We found that the TRPV family is involved in the production of Wnt5a as a mechanosensor. The other mechanosensor candidates are focal adhesions. To investigate the involvement of focal adhesion kinase, FAK, in Wnt5a production, we applied the stretch stimulation in the presence of FAK inhibitor, PF. The stretch stimulation increased Wnt5a messenger RNA, but the enhancement was attenuated by FAK inhibitor in a concentration-dependent manner. In addition, to investigate the downstream of FAK, we examined the phosphorylation of AKT and ARC using the Western blotting. FAK inhibitor decreased the phosphorylation of AKT and ARC. This is a summary of the part 2. We found that the mechanical stimulation activate integrin FAK, as well as TRPV channels. The increased intracellular calcium augments the Wnt5a messenger RNA expression through PI3 kinase AKT and MEK1/2 ARC pathway. Then I'll go on to the last part. To understand whether mechanical stimulation could affect neurite elongation indirectly through Wnt5a protein released from rat periodontal ligament cells, we evaluated the neurite length. The TG neurons, trigeminal ganglion neurons, were dissociated from 8-week-old mice and were cultured in the supernant media of the RPDL cells. with or without the mechanical stimulation for 48 hours. Trigeminal ganglion neurons have the very short neurite in the medium from non-stretched RPDA cells. However, trigeminal ganglion neurons have long neurite in the medium from stretched RPDA cells. We measured the neurite length and we summarized the neurite length per cell. The neurite was 17 times longer in the medium with stretch than in the medium without stretch. To confirm the Wnt5a as the elongation factor, we added IgG as a control or anti-Wnt5a antibody, neutralizing antibody for Wnt5a to the medium with stretch. IgG had no effect on the neurite elongation in the medium with stretch, but anti-Wnt5a antibody attenuated the elongation of mouse trigeminal ganglion neurite. The results are summarized in the bar graph. The neurite length decreases to 25% by anti-Wnt5 antibody. These results suggest that Wnt5a released from PDL cells elongate TG neurite. Wnt bind to receptors such as frizzled, lick, and roll. After binding of Wnt, these receptors various cellular responses are indicated, such as bone calcification, proliferation, and migration. These responses are induced after binding of Wnt to these receptors. According to Dr. Horigane's report, when Wnt5a binds to Lick receptor, axon outgrowth is induced through Kinase pathway. We investigated the localization of Lick receptor in primary cultured trigeminal neurons using confocal microscopy. Double immunohistochemical staining using anti-tubulin beta-3 antibody and anti-Lick antibody demonstrated that Lick receptor was expressed on the plasma membrane of soma and neurite, especially the growth cone and branching point as indicated by arrows. These data suggest that Wnt5a elongates neurite via the Lick receptor. Here is a summary of our result. The mechanical stimulation is received by mechanosensors, integrin, FAK, and TRPV channels at the plasma membrane of PDL cells. Activation of the mechanosensors is followed by synergistic phosphorylation of AKT and ARC. These pathways potentiate Wnt5 expression. Thereafter, Wnt5a released from RPDA cells enhance the growth of neurite in primary cultured mouse TG neurons through the binding to the Lick receptor. These experiments are mainly done by Dr. Kaori Takahashi as her thesis work. Thank you so much for your kind attention. Okay, now let's open questions and comments from the audience.
May I have questions?
Yes, please.
Thank you very much, Dr. Wakamori, for your presentation. It's very nice and it's very informative, a lot of data there. My question is that actually we do the same kind of experiment, but we use compressive force applied to the PDL cells. And from sequencing analysis, we found that compressive force can induce a lot of Wnt ligand expressions. Wnt5a also include as one of the upregulated Wnt gene as well, but there are other Wnt ligand, for example, Wnt2b, Wnt11, Wnt7a. So are those Wnt ligand involved in the mechanism how PDLs control the neuron as well, besides Wnt5a that you study in the current presentations?
Thank you for your question. We have to do more experiment on another Wnt5, but today we cannot show the data. We have to do more experiment. And to other factors also in some today we don't show the data, but some factor increase, but we did not show the physiological role of that factor today. We need more experiment.
Thank you very much.
Other questions? I welcome you comment as well. Okay, let's move on to the next speaker. The next presentation is about the paper entitled Mechanical Force and the Control of Periodontal Ligament Cell Behaviors. This paper will be presented by Professor Sanafam Osasanon from Chulalongkorn University, Thailand. Please.
Good morning, all delegates. It's an honor for me to be here today to present my research in the International Symposium for Interface Oral Health Science 2022. My topic will be entitled Mechanical Force and the Control of Periodontal Ligament Cell Behaviors. Periodontal ligament is a tissue that locates between tooth root and alveolar bone. It supports tooth to stay in the bone. Not only physical support, in the same time, it performs biological function in regulation of tissue homeostasis for the periodontium. Periodontal tissue subject to the force all the time, for example, for chewing, biting, or even speaking sometimes. The tongue touches teeth and leading to the force to the periodontal ligament tissues. So the role of force in periodontal ligament cannot be excluded. This study is from 20 years ago from Kaneko et al. Published in Journal Periodontal Research, showing that when the teeth that not subject to the force, so it have occlusal hypofunctions, the periodontal ligament was changed not only in terms of morphology but also the component as well. As you can see here, the control periodontal ligament have a little bit wide space, but once the teeth subject to occlusal hypofunctions, the periodontal space is decreased by time. Not only that, the orientation of periodontal ligament is disoriented when the teeth are not subject to the force. And as you can see in the lower panel here, when they stain for chondroitin sulfate, which is glycosaminoglycan, see showing that the teeth that have occlusal hypofunction have reductions of chondroitin sulfate expressions. So from this result, implying that the mechanical force have a very crucial role in terms of control or maintenance periodontal ligament functions. how force regulates cells behavior is have many factors and parameters that has been considered. For example, direction of the force that apply to the cells, type of the cells, duration, magnitude, and the times. All these parameters affect the response of the cells to the force. Our lab studied the role of mechanical force in various types of cells, for example, osteoblasts, dental pulp cells, stem cells in both pluripotent and mesenchymal stem cells. And one of the cells that cannot be omitted is the periodontal ligament, which is subject to the force all the time. So the role of force in the control of biological functions of PDL cells is indeed very important. Our previous studies in other cell types, for example, in stem cell isolated from human exfoliated deciduous teeth, and also the cell isolated from mandible, the osteoblast, showing that mechanical force can control many various functions of the cells, for example, maintaining stemness in chets and also induce differentiations of human mandibular derived osteoblasts. In chet cells, when we apply mechanical stimulations, it induces IL-6 expressions, and that will further signal to the cells through the JAK pathway and induce ATP release, and that ATP will result in the induction of REX1, which is a pluripotent stem cell markers. So all these processes showing that mechanical stimulations can apply to the stem cells and promote stem cell maintenance. However, in osteoblast cells that derive from mandible, when we apply the intermittent compressive force to the cells, it causes the release of ATP, and this ATP binds to the P2X1 receptors and cause upregulation of Wnt3A, which further influence the oxalic expression with the key transcriptional factors in orthogenic differentiations. So all this result from our previous study showing that different type of force, different type of cells, how manipulate the force to the cells, for example, force magnitude, force type, durations, time, can induce different functions of the cells. So in this study that we study in periodontal ligament cells, we select compressive force because during chewing or analytic force applications or in some traumatic occlusions, the compressive force plays a major role in the periodontal ligament tissues. There are various models that can be used to apply compressive force in vitro, but our study we used this model in the figure D. It's called hydrostatic pressure generated compressive force by piston and cylinders. So when we operate it, the machine will press down at the culture medium, forcing the pressure to compress to the cells, the culture on the bottom of the plate. And this model we published, we used in Developmental Research in 2015. So we use compressive force, but we try to alter one parameter, which is the how to apply the force to the cells. We apply continuous or we apply it intermittently. So all these parameters we set to be the same. For example, we use the same magnitude, we use the same durations of the force, we use the same type of the force, but we alter how or method to apply the force to the cells. As you can see here that when we do RNA sequencing analysis to examine the global gene expressions between PDL cells that subject to the continuous compressive force compared to the intermittent compressive force, we found different dysregulation of the genes. Approximately about 500 genes are dysregulated in continuous compressive force treated periodontal ligament cells. However, about 2,000 something genes are dysregulated in intermittent compressive force treated PDL cells. And as you can see here is an enriched pathway in both continuous compressive force and intermittent compressive force. To further determine whether the force application have anything to do with orthogenic differentiation of human PDL cells, we further subject the cell to the force application for 24 hours in serum-free culture medium, and after that we omit the stimulation and maintain the cells in oxygenic induction medium for 21 days. At day 21, we stained for mineral depositions, and we found that in the intermittent compressive force pretreatment, we found the upregulations of mineral depositions compared to the control, while those cells that subject to continuous compressive force did not have the significant upregulation of mineralizations compared to the control. So from this figure, we conclude that intermittent compressive force applications can induce osteogenic differentiation in periodontal ligament cells. From our previous study, we're showing that the ICF induced osteoblast differentiations in osteoblast cells by the releasing of ATP, and that ATP binds to P2X1 receptors and cause the upregulation of Wnt3A, and that Wnt3A drives the osteolytic expression and further promote osteogenic differentiations. So this is a mechanism that we prove in osteoblast cells. So we will determine whether the mechanism that occur in PDL cell is similar to those previously reported before in osteoblasts. Investigate whether when we apply force to the PDL, ATP is increased or not. As you can see here, when we apply both continuous and intermittent compressive force to the cell, both applications can upregulate ATP release. However, when we treat the ATP to the cells in various doses ranging from 0.1 to 100 micromolar, this ATP adding into the osteogenic induction medium did not have any effect on osteogenic marker gene expressions and also mineralizations. So suggesting that the ATP that released from the force applied to periodontal ligament cell may be not involved in the mechanism how intermittent compressive force induce osteogenic differentiation in periodontal ligament cells. Our study here also showing that when periodontal ligament cells exposed to intermittent compressive force, it can show the upregulation of osteolytics, which is a key transcriptional factors for osteoblast differentiations in both mRNA and protein level. And this force-induced osteolytic expression in PDL can be inhibited by pretreat cells with cycloheximide, which is a protein translational inhibitors, or pretreat the PDL cell with monensin, which is the vesicle transportation inhibitors. So this results implying that the induction effect of intermittent compressive force induced osteolytic expressions may be required the other intermediate molecules. So to further determine which is the intermediate molecules in these studies in PDL cells, we went back to the sequencing analysis and we found enrichment pathway. We tried to identify which pathway may be of interest. So we identified two pathways, TGF beta signaling pathway and Wnt pathway. So first we identify the heat map to see which TGF beta ligand that upregulate using the force applied to the PDL cells, and one that we observe is TGF beta 1. We confirmed that in the wet experiment, and as you can see here that when we apply intermittent compressive force, it significantly increased TGF beta 1 expressions compared to control, while the continuous compressive force did not have any effect on TGF beta 1 expressions. However, in TGF beta 2 and 3, both continuous and intermittent compressive force can reduce the expression of these two genes. The upregulations of TGF beta expression can be also confirmed by the protein expressions using immunofluorescence staining and also ELISA's assay as well. To determine the role of force-induced TGF beta expressions in both protein and mRNA level, we also confirm again that intermittent compressive force can upregulate TGF beta expressions, and this can be inhibited by monensin in NIA level, but not by coeximide in mRNA level. But for protein level, both chorecystine and monensin can inhibit the upregulations of TGF-beta1 expressions when the force applied to the PDL cells. To further investigate whether intermittent compressive force induced osteogenic differentiations in PDL cell while the upregulation of TGF-beta pathway, so we perform immunostaining of ostrix. Again, when we apply the force to the PDL cells, the upregulation of phthalate expression is observed in the PDL cells. And this induction effect can be inhibited by adding HB431542, which is the inhibitor of TGF-beta receptor, or adding TGF-beta neutralized antibody into the culture medium. So on these conditions, we confirming that the upregulation of OSX by force is occurred to the TGF-beta pathway. Further, we would like to determine whether TGF-beta pathway involved in the inductions of mineralization by intermittent compressive force in PDL cells. So we treat the cells with inhibitor first and then subject the cell to mechanical simulations and subsequently maintain in osteogenic medium for 21 days and stain for mineral deposition using alicylate staining. As you can see here, inhibitors of TGF-beta receptor can attenuate the effect of intermittent force-induced mineralization in PDL cells compared to the control. However, the reduction is not markedly observed. But when we inhibit the cells with chorecystine and also monensin, this completely blocks the upregulation effect of force-induced mineralizations in PDL cells. So it's slightly suggesting that although TGF-beta pathway is involved in this process, but it might be other molecules as well that participate in this intermittent force-induced mineralization in PDL cells. So that other molecule that we think might be Wnt pathway, which also dysregulate in ICF-treated PDL cell as well as determined by RNA sequencing analysis. So we went back and do the heat map to see which Wnt ligand that upregulate in the intermittent force-treated PDL cells, and we found a lot of Wnt ligand that upregulate, for example, Wnt2, Wnt5a, Wnt5b, Wnt7b, Wnt9a, Wnt10b, and Wnt11. And interestingly, all these Wnt ligand may be related or involving in non-canonical Wnt signaling pathway. We try to identify the relation between Wnt pathway and TGF-beta pathway, see which one is the first one to regulate the others. So we treat the cells with TGF-beta inhibitors prior to subject to the force applications, and we found that TGF-beta receptor inhibitors can inhibit the effect of intermittent compressive force upregulated all the Wnt ligand in these PDL cells, confirming that the Wnt may be the downstream pathway of the TGF-beta. When we inhibit the Wnt pathway using DKK1, which is the canonical Wnt inhibitors, and also IWP2, which is the Wnt transportation inhibitors, all these inhibitors cannot attenuate the effects of intermittent compressive force-induced mineralization in PDL cells. So implying that the Wnt pathway may be not involved in the regulation of osteogenic differentiation by intermittent compressive force in periodontal ligament cells. To summarize our study that I present you today, the intermittent compressive force can induce the ATP release in PDL cell, just like those observed in osteoblasts. However, the ATP release in this PDL cell did not have the direct effect to induce differentiations of PDL cell toward osteogenic differentiations. However, ICF or intermittent compressive force treatment caused the expressions or release of the unknown molecules which we not know yet, and this unknown molecule will induce the upregulations of TGF-beta 1 in both mRNA and protein level. TGF-beta1 then further bind to receptors and cause the activation of TGF signaling pathway and upregulate the oxalate expression, which further drive the oxygen differentiation in periodontal ligament cells. However, it should be noted again that when we adding TGF-beta receptor inhibitors, although it can attenuate the effect of intermittent compressive force induced mineralization in PDL cells, but the attenuation effect is not that marked difference. However, when we inhibit with monensin and coenzyme, the effect of intermittent compressive force induced mineralizations in PDL cell is much abolished. So implying that there may be other intermediate molecules that play the roles in how intermittent compressive force induce oxygen differentiation in periodontal ligament cells. And lastly, I would like to thank all my funding agencies, especially JSPS and Raku PhD program that offer the scholarship for Dr. Jinamano Gomincho, who mainly involved in this study I show you today. And also I would like to thank all my collaborations at Tokyo University, Professor Hiroshi Ekusa, Assistant Professor Kunsuk Lim Laksasin, Assistant Professor Hiroko Okawa, and also for the folks there in University of Paris that we have collaboration a lot lately. So thank you very much. I'm ready for the question if you have some. Thank you.
Professor Wakamori, you are muted. We cannot hear you.
So, sorry. Thank you very much for your excellent presentation. And now let's open questions and comments from the audience. Okay. No chat. Okay. Okay, so time is coming. Let's move on to the last speaker. Thank you. Professor Spur. The next presentation is about the paper entitled Root Cement, the biological interface between periodontal regeneration and periodontal repair. This paper will be presented by Professor Alex Spur from the University of Sydney, Australia. Please.
Thank you very much and good day. And thanks a lot for this kind invitation to speak on your amazing conference. And I start right away. So I would like to talk about the importance of root cementum as the interface between regeneration and repair. And we see root cementum as one of the key tissue for periodontal regeneration. When we talk about periodontal regeneration, we have a look what tissues we need to regenerate. More or less, we talk about 3 different tissues. We know that periodontal tissues consist of 4. We have the gingiva, we have the root cementum located on the root surface. And then we have the periodontal ligament, what we have just heard before, which is filling the gap between the root and the bone. And then, of course, you have the alveolar bone. We don't regenerate gingiva. Gingiva, we usually in the clinic we transplant, but we have to regenerate cementum, ligament, and bone. And interestingly, when we look on the fetal development and what you can see on the right-hand side, that's a histology of a rat fetus. You can see the primary oral cavity, and that's the enamel organ of an upper central incisor. We know that this condensed ectomesenchymal tissue is more or less the stem cell population that's responsible for the formation or development of root cementum, ligament, and bone. So when we look at this root cementum, and we spent quite a couple of years ago to study root cementum development, we know that there are a couple of cells involved. We know we have, regarding the formation, the cementoplast is more or less the crucial cell line, but we also know that there are obviously some specialized fibroblasts involved, which also derive the dental follicle or dental sac. Cementocytes are metabolically inactive. They are not dead, but metabolically inactive. But what we know is they could be to some kind stage reactivated. But let's talk about the cementoblast and the specific fibroblast. So when we think about periodontal regeneration and particularly root cementum, it's getting even more complicated, because when we look on the root surface, there are unfortunately different types of root cementum. And when we start from the cementum number junction, the first type of root cementum we approach is called AAC, or afibrillar cellular cementum, which is this part what you can see here. This is enamel, that's dentin, and this is the cementum and the ligament. This is more or less the product of a dying ameloblast layer during the tooth development. Doesn't really have any functional importance. When we go a little bit further down towards the apex, and we are now in the coronal 2/3 of the root surface, we approach another kind of cementum, and that's called the cellular extrinsic fiber cementum. And this is a very important cementum, and I will tell you why. Because what you can see, first of all, the characteristic features of the cementum, there are no cells enclosed. The cells are more or less on the surface. And you see a quite impressive fiber content embedded into the cementum. You see the typical structure of collagen fibers, and what's very important is the fiber density. Because the fiber density in the particular areas of the cementum tells you something about the importance and the function of the cementum. So if we go now further towards the apex, and now we are in the apex area, or apex is third, or also in the vocation area, this is a kind of different cementum. What you can see here, it's a thicker layer of cementum. You see all these small dark spots that are cementocytes embedded. And what's also quite obvious compared to the previous one, you see a significantly lower density of the collagen fibers. So the question is now again, when we talk about regeneration, and of course the question is what kind of cementum would we like to regenerate? And regarding function, I mean, it's quite obvious that we would like to regenerate the cellular extrinsic fiber cementum. There's another one which I just forgot, and that's the cellular intrinsic fiber cementum. This is the one which is only a repair cementum. It's usually just the job is to cover the exposed dentin surface and avoid further resorption. So, but what we would like to get is this one. That's probably the most, the tooth or the root would benefit most if we would be able to regenerate that kind of cementum. And what we know that this cementum seems to be formed by a specific type of fibroblast from the dental follicle. What we've also seen before is that the ligament, of course, the next tissue is a very important one, and we know that in this tiny space between the bone and the root surface, we have all types of cells involved for every tissue that's in this area located as well. And we also know that the cells that have the capacity to regenerate the whole periodontal tissues, the so-called progenitor cells, are located in the ligament as well. And it seems what you can mark these cells, they seem to be located around the blood vessels. And we know from some studies that if necessary, obviously, there are further stem cells that can be recruited via the bloodstream. But they are not just cells, of course, what we have seen before in the nice lecture before, they also have ligament fibers. And you know, they are running in all kinds of directions. And that's another important question in function-wise from a clinician's perspective. What's the fiber orientation we would like to achieve if we have a fully regenerated periodontal tissue? And of course, function is important. And last but not least, then we have the bone. And the important part for us is this lamina cribriformis, so now they call it the bundle bone. This is just this 0.1 to 0.4 millimeter thin bone, which is more or less dependent of the root cementum. In the same way, like the root cementum connects the fibers to the root surface, the bundle bone connects the fibers to the jawbone. So this is what I call a unity. It's a structure, cementum, ligament, and this bundle bone, and that's what we try to regenerate. If you look on the developmental stages, and that's very important, we can learn a lot from the developmental tissues, from development. We know that once the ligament or the periodontal tissues are formed and the root formation starts, the first tissue formed is usually root cementum. That's the first that shows up. Afterwards comes the periodontal ligament, and then with with the beginning of the eruption of the tooth, you see that the bone grows more or less in a more coronal position. And this was the reason why we said, okay, it seems that root cementum is the key tissue. You won't get a periodontal regeneration without root cementum, that's for sure. And then we have a look on what's on the market, what is used these days for periodontal regeneration. We know that in some studies, and we tried the same, they tried use of bone substitute, bone graft. If we looked on the histology we could achieve, it wasn't very successful. We never really got the formation of a root cementum. If there was a root cementum, it was just this repair cement that was not really firmly attached. The other problem is we saw some root resorption. When we looked at different growth factors, we tried several of them. Unfortunately, particularly BMPs, we showed a massive root resorption, so we couldn't use them. And then, of course, we have the guided tissue regeneration, membranes. And then what interested us most really was the enamel matrix proteins. And this is what I would like to talk today. And when we started this project, it was a few decades ago when I worked in Sweden at Karolinska, and we tried to compare what happens, what are the crucial developmental stages during the normal development of the periodontium, and what happens when we try to regenerate. And if you look at these stages, we see right away from the beginning that in normal, when the periodontal tissues develop, it starts with the breaking of the Herdwick's epithelial root sheath. If we try to regenerate, it doesn't start with the root sheath, it starts with the formation of a blood clot or blood coagulum. So what we recognize as a quite different starting point. Once this has formed this clot, and when the herd sheath breaks up, we see that what happens is a reorganization of the cells between of the tooth and the bone during development. And when we try to regenerate, we see the granulation tissue form. So there's still quite a difference, but the interesting part is from now on, it's quite similar. So what we see now is that, first of all, there are progenitor cells present or migrate into the defect or into the space. Then there's an expression of different orientation, differentiation factors, and then the progenitor cells differentiate into cementum-forming cells, ligament-forming cells, and bone-forming cells. So the question was, what can we need to do to change the starting point in a way that we don't end up in a granulation tissue, but we end up more or less in a regenerated periodontal complex. So now, why did we try enamel matrix proteins? Because enamel matrix, everyone knows, is part of the enamel organ. It's more or less the protein matrix that's then replaced by the enamel, the mineralized enamel. What's the point of using these proteins? The interesting part is what we recognized quite early when we did some immunohistochemistry studies, that there are different enamel matrix proteins that are expressed during root formation and particularly are involved in the cementum formation. And one of the major studies was done by Hammerström and Linskog, and what they recognized was when the root formation starts, and if you look on this graph, you see these are pre-odontoblasts and odontoblasts, you know, they form pre-dentin, and then the dentin starts to mineralize, and the just mineralized first amount of dentin is what we call the mantle dentin. And once this mantle dentin is mineralized, then the Herdwick root sheath, which is displayed by this bluey cell, separates more or less from the root surface. It breaks up and it undergoes apoptosis regulated cell death. But what Hammerström also recognized was that before the root sheath cells leave the root surface, they secrete a specific protein layer on a mineralized mantle dentin. And then what happens afterwards, and this is what you can see in the real picture there in histology, once this these matrix proteins are secreted on the root surface, then cells of the surrounding tissue migrate onto the root surface, a monolayer, which forms alongside of the mantle dentin, and these cells differentiate into cementum-forming cells. Looks like fibroblasts, and then the beginning of a cell extrinsic fiber cementum starts. So then, once you recognize that seems to be a connection between these different proteins that are part of the matrix and the cementum formation, we started with some studies. And we first of all was a baboon studies, and we did two models. First was an extraction model, where we extracted the lateral incisors, we drilled more or less a defect or a hole onto the root surface. We applied enamel matrix from pigs, and we replanted the root into the former socket. And what we could see that with the application of the enamel matrix, you could see the new formation of cementum, whereas on the control side, where we didn't apply any matrix proteins, there was a root resorption. So that's quite interesting, but it doesn't tell much because that's far away from a clinical scenario. So the next stage was then what we call the buccal dehiscence model. So what we did, we removed more or less the buccal bone from the first premolar to the mesial root of the first molar on the buccal side, till about 4 to 5 mil above the apex. Then we marked a small notch in this area, and we removed with a high-speed handpiece the whole ligament and cementum. So what was left after the surgery more or less was root dentin. Then we applied again the enamel matrix on the surface. On the test side, on the control side, we did the same surgery, but we didn't apply anything. So that's how it looked like. You can see the first premolar, second premolar, and mesial root of the first molar. So an outcome was quite interesting because what we recognized was then on the side with the enamel matrix, about after the first few days, 7 to 8 days, we saw again the migration of fibroblast-like cells forming a monolayer on exposed root surface, and the rest was more or less granulation tissue. A little bit later, another 1 or 2 weeks, you could see now cementum formation has started. You could also see the first fibers of periodontal ligament extending. And interesting-wise, we saw different spots of bone matrix formed. It wasn't growing just from the apex. We saw these spots more or less parallel on the whole root surface. And even in the early bone matrix formation stage, we recognized that it was quite exposed already and connected to the fibers. A little bit later, after 8 weeks, you can see this was the notch. So the bone was removed to let path. You see now new cementum formation, ligament formation, and bone formation. And interesting was also that the dimension of the ligament space was maintained. So where we placed this notch or we drilled the notch, you can see we had this kind of nose formed in towards the root surface. So it maintains the same ligament space. And this picture shows more or less all the stages that really happens. Again, you can see the notch. So this is the old bone. It was removed at that point. Now you can see this is old cementum, the more purple-like one. And you can see this has new cementum formation, which is growing around. And then with the new cementum formation, the ligament formation started. And the ligament fibers got longer and longer and longer. And afterwards, more or less, the bone followed. So what we achieved was more or less, and this was the type of cementum we could achieve. It's a cellular extrinsic fiber cementum. So what we achieved more or less was very similar to what we see during the normal development of the periodontal tissues, right? The first tissue formed with cementum, then followed by ligament, and then followed by bone, and that's why we call this then biomimicry. So then the problem was we tried to explain what's really happening. We see what's going on histologically and clinically, but of course you want to know what's going on. So the next stage was then we used the Spectral rats at an age of 6 to 7 days. This is when the first molar in the mandible, the crown is more or less growing, but there is no root formation. And the crown, the enamel organ is still in the secondary stage. So what we did, we made a lateral incision. This is the muscle. You see the arteria facialis. We opened the fascia, shifted the arteria and the muscle to a more coronal position, and we opened the window in the mandibular bone. And underneath grows the developing first molar. So we took the molar out. This is what you can see. Dimension is about 1.5 to 1.8 mil, and you see the cusps. So what you see, this reddish tissue is the enamel organ. So now we used minor curettes and we stripped the enamel organ off. The idea was to expose this freshly secreted enamel matrix. It's a soft tissue, it's not hard. So the crown, it's still a lot of enamel matrix, hardly any mineralization. So we stripped the enamel organ off, exposing the freshly secreted enamel matrix. And then we turned the crown in an upside down position, and we replanted the crown with the tips downwards. And then we closed the hole again with a window. We preserved the bone in the cell culture medium. And then we waited, and what we saw after 8 weeks is that on the crown we saw a cementum formation after 6 to 8 weeks. This is between two cusps. Again, you can see a cementum formation there and there. And these are separated enamel matrix pieces. They showed as well some cementum tissue formation. And then afterwards, we recognized about 10 to 14 days, the ligament fiber started, and afterwards came the bone. So we generated more or less complete periodontal complex around the replanted crowns. So now the next question was, is that dependent on the cell type, or does that happen all the time? So now we did the same surgery again, but we didn't replant the crown in an upside down position in the form of socket or alveolar. We now transplanted this crown into the subcutaneous position of the neck of the rats. And what we saw was just nothing. There was no cementum-like tissue formation. We saw just again this is dentin, enamel matrix. We saw even a resorption of the enamel organ, but we never saw a cementum formation. So the issue is not that enamel matrix alone doesn't do much. You also need specific cells that are capable really to conduct the kind of regeneration. So then the next step was we tried to find out what's really happening in these cells. So we used different deoxygenin marked probes, mRNA probes. And what we recognized was that the first cells that were seeding on its exposed enamel matrix significantly upregulated really their mRNA for collagen type 1. The cells that are not in direct contact with this matrix, nothing really happened. So it was clearly a cell matrix interaction. So the cells have to be in contact with this with the matrix. We also did some cell culture studies where we took extracted teeth, and you can see the cementum, the old cementum. We applied the matrix protein. That's how it looks like. They form this globally. And then we submerged this in a cell culture medium with PDL cells, and the PDL cells were growing on the matrix proteins and start to form some mineralized tissue. The interesting part then, when we went into some clinical studies on human patients, you also recognized that the healing was very well. There was hardly any inflammation. We never had a problem with infection. So we did some other studies where we co-cultured specific type of matrix proteins with the specific or different periodontal pathogens. We used Porphyromonas gingivalis, Aggregatibacter, Prevotella, Treponema, everything. And what we recognized was that particularly regarding P. gingivalis and Prevotella, there was a significant inhibition on the growth of these periodontal pathogens if we co-culture them with specific enamel matrix proteins. So there seems to be an antimicrobial effect as well. Then we did a large research project. It was funded by European Union with different countries, and this was a study that was done by a colleague of us in Leeds, Professor Robinson, and they co-cultured again the enamel matrix proteins with PDL cells, and what they recognized was that the cell seems to ingest the enamel matrix proteins into the cytoplasm, and then they transport the enamel matrix protein somehow close to the nucleus, and then the gene expression seems to change. Some other partners of this project also did some receptor studies, and they recognized that particularly PDL cells have specific receptors for these specific enamel matrix proteins. So, and then of course, what was also convincing about the clinical data that whenever and this was one of the studies we did, we see all of the patients were only treated with enamel matrix protein, no membrane, no bone graft. And you see this how it looked before. This was after 12 months. Again, that was a 12 millimeter defect before surgery, and this was after 12 months. Again, this is after 12 months. This was a young patient. She was 15 years of age, and that's again only treated with the matrix proteins, and this was the outcome after 12 months. So the outcome was quite impressive. And we saw histologically, it was really the complete regeneration of all tissues. Another advantage for clinicians is when they use matrix proteins, they're easy to apply. And the only issue is it's advised that you create a free fiber network for the proteins to attach. So what we usually do, we apply an EDTA gel which is a shellate formation, so it more or less removes all the smear layer from the connective tissue fibers of the root surface. We leave it for about 2 minutes and then we rinse it. So we have a 3 fiber network, like a small lawn. And then afterwards, once we rinse it away, we apply the proteins right away. And it's very important what we recognize, that once you apply the proteins, there shouldn't be any saliva contamination, and particularly no blood contamination. That's an important part. And then you just close it. This is it. And we also used it for recession coverage. This is one of the cases we did a lot. We published as well. You know, there was a connective tissue graft together with matrix proteins. And this is the case 12 years later. So we achieved quite a significant and stable outcome, and what the study results showed, that the stability significantly improved. The funny thing is, if you look on the matrix, there's a huge bunch of different proteins, and also we noticed, because of the alternative splicing mechanisms, that from the same gene there are different proteins. So what we also checked the effect of the involvement of some of these proteins in the bone formation. We found some of the proteins in intramembranous bone formation. That's, for example, in the red features of the skull, you can see the immunochemistry. This was a vertebra endochondral bone formation, and you see again the same protein expressed in the matrix, in the cartilage matrix. This is a growing gene, and we recognize the expression when the pre-osteoblasts differentiate. And once the differentiation is over, the bone is formed, then there's not much expression left except the filibriosteum. And we did an mRNA histology too. We started also to use some of the proteins then to regenerate the bone, and we used them to apply them. That's a bit tricky. We created some inserts where we embedded this matrix protein, and we cemented the insert more or less into the bony cavity with this matrix and the proteins, and you see then the bone grows towards the insert. After a few weeks, the insert breaks down, releases these proteins, and then you have further growth till the bony defect is more or less completed. So what we recognized was that, yes, really the cementum seems to be really, from the clinical perspective, the key tissue for periodontal regeneration. If you are not able to regenerate root cementum, you will never get periodontal regeneration. And it's also important, if you regenerate bone and the bone grows towards the root surface, then there's no cementum or ligament formed, you know, we get root resorption for root ankylosis. And that's why we see root cementum really as the crucial interface between regeneration and repair. And again, thank you for the invitation, and thank you for the opportunity to talk on your great conference. Thanks a lot.
Thank you, Professor Spur. Thank you very much for your excellent presentation based on basic and clinical aspects. Now let's open questions and comments from the audience.
May I have a questions for you?
Yeah, please.
Thank you very much for your presentation. It's very nice. Actually, I came across your paper last year in Geomaterial Science about the porcine ECM. That's a very interesting paper anyway. Okay. But anyway, my question is that when you present today, you use EDM alone without any bone graft and membrane. And it seemed that the effect, although it have some regeneration, but it's not that like completely regeneration or something like that. Would you suggest to combine those, I mean, EDM and other bone graft as well to enhance the effect on regenerations?
That's a good question. Actually, yes, you always get all three tissues. That's what we see, except when we did this recession coverage where the buccal root surface was completely without bone, and there was some histology. There was less bone formation. That was more or less cementum and ligament attached. But when we did the study, the long-term coverage success rate was always better with EMD than without. And the reason was, without EMD, it was a long chunk to the freedom, there was no attachment. And the bone formation was a bit like a wheel. It was about to close. In intra-osseous defects, I think that's what you're interested in. Still, if it's a narrow defect, I wouldn't use any bone graft. It seems to be more, it's an inhibitory factor. But if it's a larger defect, and my law of the thumb is usually once the crestal bone is more 3 mil or more away from the root surface, then I would apply the matrix on the root surface and then fill the rest with the bone graft, just as a scaffold, exactly what you mentioned to promote more or less the bone formation a little bit, yes. But in narrow ones, I wouldn't.
I see. Thank you very much.
No, you're welcome.
Okay, time is coming. I would like to thank both the speakers and the audience for providing such an interesting session and discussion. This session is now closed. Once again, thank you so much for taking time to join a Keynote 5 session. Thank you.
Thank you. Thank you, Professor Num and Professor Axel. Thank you.
Thank you.
Thank you very much. This concludes the morning program. We will have lunch break from 1:10 p.m. E-poster session will be started on following webinar indicated at the chat box. Thank you. And the main session will be started from 2:30 p.m. on time. Thank you very much.